Well, thank you for making it today. And that was a rough commute this morning. Supposed to end at 2. We'll see. And then warm up to 50 by the end of the week.
That'd be nice. I saw a bunch of Robinsons last.
Yeah.
It's like, they're probably hating it.
Yeah, every time I saw a robin fly by, I'm like, oh, I'm sorry. I came a little early.
They with all the geese. Y'all are your way.
Oh, the geese are here too. I didn't see that so much.
They were just put on the.
Oh.
They were in.
Hang in there. Yeah.
Yeah. Yep. My theory is those ones are the ones who did their first migration last year.
And it was when it was so nice.
Warm earlier.
Yeah.
And now they came back too soon. They're like, oh my God.
Yeah. Well, we'll make it through. Soon it'll be 50s and it'll all melt.
Yay.
Don't forget the SVP assignment due Wednesday, 11 a.m. So make sure to get that. And then I'll be posting a gymnosperm one, probably with the same parameters where it'll open on maybe even Wednesday, we'll see, but something close to that due next Wednesday, probably. So I'll be posting that soon. Okay, so we got to get into the gymnosperms again and make some progress on thinking about their life cycle. They have different life cycle compared to what we've covered in the other groups. So remember, we have the sporophyte is the dominant phase. That hasn't changed. But a huge change is that the gametophyte is going to be held onto. So the sporophyte is going to hold on to the spores, and the spore will develop into the gametophyte on the sporophyte, especially when we look at the megagametophyte. And so everything's being held and protected, and everything's getting reduced, fewer cells. And so let's go to that idea. So we covered this last time where the megaspores, remember everything's heterospores. We have megaspores, we have microspores. The megaspores get held onto, they don't get released. They stay in the megasporangium, and there's only one. It gets reduced down to just one megaspore. Sometimes it's called a megasporocyte or the megaspore mother cell. It's just one. And then that one needs to go through meiosis, which you end up with 4 cells, but 3 die away. So you're just left with one functional megaspore. So now that megaspore, so it's all retained there in the megasporangium. Now that megaspore has to divide, grow, and develop into the megagametophyte. And so that functional megaspore is going to go through a bunch of mitosis. So here's a close-up of the functional megaspore inside and retained inside the megasporangium. Now this functional megaspore is going to become the megagametophyte. And so it looks kind of crazy and haphazard early on, but eventually you'll see that things become more tissue-like, and you'll get the archegonia showing up, and 1 egg in each of the archegonia. And then there's a tiny opening called the micropyle so that the pollen can get in with the sperm. And we saw that, yeah, okay, technically all eggs can be fertilized, but usually only one develops. So that one wins and keeps developing. So this is after that megaspore went through, cell division, mitosis, and developed into the megagametophyte. So that was where the megaspore was. Now it's developed into the megagametophyte. And you can see the archegonia and the egg in each one, big fat egg. All right, so that was once the megaspore. It's still withheld within the megasporangium. Okay, so it's still tucked away there. And then this integument tissue is further protecting. So you can see how precious these eggs are. Everything's getting withheld and protected, except you have that little opening, the micropyle. And you typically do see a pollination droplet, which will catch the pollen grains that get released from the male pine cone or the pollen cone. Okay, so that integument eventually will actually become the seed coat of the seed. We'll see that. in a little bit here. You with me so far? Questions on this? So there's one megaspore mother cell.
So the archegonium is in the megasporangium?
Yeah, so everything gets tucked away in the megasporangium. It's weird. Because usually we say, oh, you know, the sporangium holds spores and those usually get released out. And that's not the case now. So the megasporangium says, nope, I am not releasing my megaspores. I'm going to reduce it just to one. And that one's going to go through meiosis and then I'll 3-die off. I'm going to have a functional megaspore. I'm going to let that grow and divide and become the megagametophyte. And I'm still holding on to it. It's never released.
Where's the megaspore at? Is it in the anchegonium or is it?
So the megaspore developed into this. So when you have the megaspore, the functional, here, the functional megaspore. Right, so this is the, after meiosis has happened, 3-die off, you're left with one. Now this has to go through a bunch of growth and development, and it's going to become the megagametophyte. So if you look at this picture, and then we go back to where you see the megagametophyte, that's where the megaspore was, and now it's developed into the megagametophyte. And it's still being held within the megasporangium, and you still see this integument tissue. So that was what we call the megaspore. But now it's the megagametophyte. And once you start seeing archegonium and all of that, then we say, oh yeah, that's the megagametophyte for sure. Other questions on that process? Yes.
Is that what the pine cone is?
Yeah, so we're gonna put it into context of the cone now, or shortly. We're gonna do that. So yeah, this is all happening in the pine cone tissue. If you look at the scales of the cone, eventually you're going to see the developing seeds sitting like right on the top of the scale if you look down on the pine cone. Okay, so now that megagametophyte has the eggs in the archegonia and is ready, you know, mature enough now where if sperm come along, they'll be good to go for fertilization. So let's assume that happened. And so then after that egg gets fertilized, here you're seeing the pollen coming along. And I'll talk about pollen and what's going on there, but the pollen is conveying the sperm and will fertilize the egg. And then that will be the zygote, which has to grow into the embryo, which is basically the baby sporophyte. And then the seed has to develop to protect. So we'll talk about that. And then the megagametophyte tissue stays there, right? So the megagametophyte tissue is actually, when you crack open a seed of a gymnosperm, we don't say endosperm. There's no endosperm in there. It's megagametophyte tissue that's actually being used as a source of nutrition. Normally, when you talk about seeds, we tend to focus on angiosperms and say, oh yeah, endosperm is what, you know, feeds the germinated seed. But in this case, it's going to be the megagametophyte. So that's a big difference compared to the angiosperm. So we're going to talk a little bit more about seed development. And when you have the fertilization event and more growth of that zygote and embryo, and then you'll see the seed develop right there. So this is all happening in the female pine cone. So let's go, I think we showed this as well last time. So here's your functional megaspore on the far left. This has to develop into the megagametophyte, which is here in the middle. And they're saying, okay, then let's, you know, think about the pollen having to get in there through the microtyle, delivering the sperm. Remember, the sperm aren't motile. There's no free water for this. And then you have the development of the embryo, which is essentially the baby sporophyte in that seed. Okay. Under the microscope, you would see something like this. I usually work from the outside in to figure out what's going on with the different pieces here of the anatomy. So I know that the integument is there, really protective. So that's what you got going on here, the integument. And then you can see the micropyle region in here. And then I know that the megasporangium holds in the megagametophyte. So that's the megasporangium tissue, and all of this is the megagametophyte tissue in the inner portion. You can see the two archegonia, two fat eggs in there, and the rest of the megagametophyte. So that still has to develop. That doesn't look like it's been fertilized yet. There's no embryo developing yet. So it's just waiting. It's waiting for the pollen to come by. So with the pollen grains, if you look at that, each pollen grain is 4 cells. That's important. I'm going to be asking you to know that because the angiosperm is different. In the gymnosperms, we see 4 cells, and that's how they get shed. So what we have are 2 prothalial cells, one so-called tube cell, and a generative cell. So it's the prothalial cell, tube cell, and generative cell. That's how it's shed. So there's no sperm yet. Okay, there's no sperm yet. So when it gets shed from the male cone, they say it's still immature. The pollen grains are still immature because there's no sperm showing up yet. There still needs to be a little bit more work before we end up seeing some sperm. So once the pollen grains land on a mature ovule with a megagametophyte and eggs in there, then eventually what we'll see is that generative cell will divide to produce so-called stalk cell and a body cell. Sometimes the body cell is referred to as a spermatogenous cell. I have that in the notes. It's hard to say, so I opt not to say it. Spermatogenous cell, which spermatogenous, sperm is in the word, because that's the cell, the body cells will divide eventually to give 2 sperm. All right, so each pollen grain eventually will provide 2 sperm, but they don't get dispersed like that yet. They have to reach the ovule. And then you get additional mitosis where some sperm develop. So they're trying to show that here on this diagram where you have the male cones, the pollen cones on the tree, on the sporophyte. And here you have the microspore mother cell or the microsporocyte. Has to go through meiosis. Okay. And so when you go through meiosis, you end up with 4 cells. So each of those 4 microspores develop. There's no 3 dying off, 1 survives. Not when it comes to pollen. As you know, any of you who are allergic to pollen, there's lots of pollen. And so all of those from each meiosis event, those 4 cells will develop into a pollen grain consisting of 4 cells. And then they'll get released like that. So here they're showing the 2 prothalial cells, the generative and tube cells. So that's how it's shed. And then hopefully it makes its way to the micropyle of the ovule. And then eventually a couple sperm will show up.
So is it 2 per cell? So overall will it be 8 sperms produced for the 1 pollen?
No,
No.
no. So the generative cell is the only cell that's going to divide to produce 2 sperm. So each pollen grain will produce 2 sperm ultimately. Yep, you got it. So this picture I'm showing bits and pieces of, I'll show the full thing. This is from your book and it's accurate. And it shows some nice pictures of under the microscope what you would be seeing. So this is good. It's a little challenging to teach off of, I think, because the words are so small. This is from a previous edition and I kind of like it for teaching out because it's a little easier to see, but they're both correct. Okay, so it's fine, whatever one you study off of, or both. So this is what I've been showing you bits and pieces of in some of the other slides. Okay, now Gymnosperms, so this is the only life cycle, by the way. We're gonna cover the 4 different phyla. This is the only one you gotta know. We're not gonna get into the details of any of the other 4 or 3. So this is the only one. And with the angiosperm, I'm just gonna tell you about one. Angiosperms go through reproduction in a variety of ways. I'm just gonna focus on one and touch on one other one. So we don't have that many life cycles left to know. Okay. Gymnosperms are a little odd in how long they take to do all of this. So I'd like to walk you through that. You don't have to write this all down word for word. It's in the PowerPoint. But basically, let's start from the point of pollination, which just means that's the definition of pollination right there in the gymnosperms. So if you look up pollination, they'll probably give you the angiosperm in the dictionary. So make sure you know this in this class. What is the definition of pollination in gymnosperm? It's the transfer of pollen grains to the ovule. So let's start from there. So let's say that's happened. About 1 month after that has happened, you get the 4 megaspores being produced through meiosis. Only one survives. So that pollen grain sitting there, you know, hopefully got delivered through the micropyle and it's sitting there on the ovule and it has nothing to fertilize yet. There's no eggs yet. Okay, so the pollen gets there and then it's like, okay, your turn. I'm going to sit here until you go through your stuff. So the female portion, you have to go through meiosis, 3 die off. You're left with that one functional megaspore. That megaspore has to grow and develop into the megagametophyte. You got to get archegonia with an egg in each one. 13 months later, you get a fully mature, well, almost fully mature megagametophyte. It takes a little while for the archegonia to fully mature with the egg. So 15 months after pollination, you finally have the female portion with the egg ready to go. So that's a long time. So essentially over a year, essentially a big, long process, very heavily protected, especially on the female side. Now, about 3 months after pollination, the pollen grain finally starts to get active. And so the pollen tube cell will start to, or the tube cell, I should say, starts to produce and germinate a long tube so it can deliver the sperm that are eventually going to show up. So it's good that the pollen lands really close to the archegonia, where the archegonia are, because they have to make it through that megasporangium tissue and get the sperm delivered right where the egg are and the eggs are in the archegonia. So about 12 months later, after pollination, you finally get that generative cell dividing and then the body cell dividing to get you the 2 sperm. So a whole year. And then 15 months, you finally have the egg ready to go. The pollen tube finally gets there. The sperm travel down the tube and make it to the eggs. Yes.
When do pine trees like lose their like pine cones? Like, is it after the 15 months, the obsession or whatever?
Yeah, I'll show you. It takes a while. Yeah. And so often, like I have a picture, a really nice picture of cluster of 3 pine cones, and you can see they're each in different stages of development. One's tightly sealed up, one's open and has seeds, one's open with no seeds, you know, so it's very interesting. I was lucky to capture that. But anyways, so let's think about this. So at 15 months, then finally the eggs are ready to go, the sperm finally get there, and hopefully get fertilization. But look at this, one sperm gets to fertilize the egg, the other sperm will die off. That's critical. You need to know that because we're going to see in the angiosperms, that's different. Okay, so this is interesting. We don't have that second sperm doing anything. It loses out. Okay. After fertilization, you get the zygote, and then you get another kind of mess of, they call it the free nuclear stage. That's the messy stage where when you look at it under the microscope, it just looks like chaos in there. And then eventually you start to see the embryo developing. So 18 months after pollination, you'll finally get a mature embryo. So it takes several months, 3 months essentially for that. And then the mature seeds typically shed out at that time. So the average is 18 months. So I'd like you to know that. They usually hedge their bets and say, well, it depends on the species. There's a lot of different Pinus species. So the genus name is Pinus. And it can range anywhere from 14 to 20 months, but 18 is a good one to remember. So year and a half before. Yes, a lot faster. And that's probably why they're the dominant plant right now.
And after that, you're ruined everything.
Yeah, exactly. The gymnosperms are like, crap, now we gotta compete with these angiosperms. So, anyways, it's a long process. And, so, when we look back at this, just remember, this is a very long process. It's, easy to look at and talk through, but this goes a year and a half in order to get it all the way to the seeds releasing out of the cone and hopefully flying away, so to speak, somewhere.
One pregnancy would take nearly as long as a baby elephant.
I know, right? And yeah, it's interesting they haven't evolved to be a little quicker, but
Yes.
yeah, I mean, they're successful still. They have their niches. So that's what the pollen looks like when it gets shed. So you have these wing cells that help with being able to stay in the air currents because the pollen grain, really, that's how they're depending, in the gymnosperms, they really do depend on wind mostly to get the pollen grain distributed. That's why there's so many pollen grains that get released. It's massive. Yeah, and then you go to your car and it's all yellow and yeah, yellow dust. So this is an interesting picture showing the female pine cone. And they're zooming in to the developing ovule right in here. This is the micropyle area. You can see the integument tissue here and here. Okay. So there's pollination occurring. You got some pollen grains coming to the ovule. They made it through the micropyle. But if you look at the ovule here, there's no archegonia. There's no eggs. All right. It's not there yet. You have the functional megaspore right there. So the pollen get there and then they're like, the female's still got to get going, and so there's a country song, Always Waiting on a Woman. I always think of that. You're always waiting on a woman. So the pollen grains are just sitting there going, okay, come on, we're going to be late. So here, this functional megaspore has to go through lots and lots of growth yet to form the megagametophyte, get the archegonia, get an egg in each one. All right, so, but they captured the pollination. Whoever did this sectioning should get the gold star for the day because that's pollination in action. That's pretty cool. So here is pretty much the same idea, except what often tends to happen is the micropyle will seal up after pollination. Okay, so this previous image, they're showing actual pollination in action. This is, okay, pollination has happened, and look at the integument further seals to protect. So again, this precious, reminds me of Gollum, a precious ring. Lord of the Rings, like precious, everything gets sealed, everything's protected and withheld.
It's like, it takes so long, you don't wanna risk it falling out.
Well, exactly.
Drop
It takes a long time. You better seal up. And so, yeah, and I'll show you some other pictures, but here's the, it's the functional megaspore. It hasn't done anything yet. Oh, the megaspore site. Oh, here, in this case, it's the megaspore mother cell that hasn't even gone through meiosis yet. So,
Does the pollen survive in there?
yeah. So this is the messy stage where it's like, what is going on here? So this is the megagametophyte in really early development. And this is what I mean by it's chaos and messy, but eventually it develops. So in this particular snapshot, they're showing just one archegonia. And all that white in there is the egg. Okay. So again, here they caught the micropyle sealed up. Right here, those are two, it looks like two pollen grains that are sending their pollen tube and digesting through. All of this is megasporangium tissue in here. So the pollen tube has to get all the way through that and reach where the egg is to discharge the sperm. So it's got a ways to go. That's why it takes, a few months to get this to happen. So the megasporangium tissue is all in here, okay, and it's surrounding. And then right inside of that, that's the megagametophyte right there with the archegonia and the big egg in there. So this is all megagametophyte. And they sealed it all up. Okay. So this is a nice image where they've put the drawing on the left and labeled it, and then you see what it looks like under the microscope. Again, this person should get a gold star, whoever did this section for the microscope mounting of this image. They're actually showing fertilization right there. So that's the egg nucleus, and that's the sperm nucleus right there. So they're actually captured fertilization. That's pretty cool. So if that happens successfully and you get good, successful fertilization, then it's a zygote, which has to go through a bunch of mitosis and differentiation to become the baby sporophyte, or, the embryo. So it's going to look kind of chaotic again and like, not much to really decipher. But eventually you'll get something like this. So this is ready to go. So all the greenish area, that's the baby sporophyte. Okay, baby sporophyte. All of the pinky red here surrounding it, that's the megagametophyte. You got to know this. Okay, this is really important because I'm going to make some comparisons between a gymnosperm seed and an angiosperm seed when we get into the angiosperms. So knowing that a gymnosperm seed is pretty fundamentally different from an angiosperm seed. That's a big thing to think about as a plant biologist. So the baby sporophyte or embryo is in here with all the green, and it's going to be the megagametophyte tissue. Once the seed lands somewhere and starts to germinate and grow, it's the megagametophyte. All that pinkish red, that's what's going to feed. It has the nutrition to support the growth of the germinating seedling. Here again is the labeling of the seed. Very nice job there. Okay, so the female gametophyte or megagametophyte, that's in this case, the purpley tissue. And then in this case, it's the green in the middle. That's the embryo, the baby sporophyte. So all of this is happening in the cones. Okay, we got to zoom in. And really, they are strobili. When you look at female cones, male cones, they are strobili because that's where the sporangium tissue is located and it's all clustered together. So that fits the definition of what a strobilus is. And so they're pretty complex, except for there's some exceptions. The ginkgo biloba and the yew family has some pretty simple cones, especially when you look at comparing what we were just looking at. You just have very simple stalks where the seeds hang out on the end eventually. So I'll show you a couple pictures here. But those are exceptions. So ginkgo and the yew family. Here's ginkgo. So it's even hard to imagine that this is a gymnosperm because look at the leaves. They look like angiosperm leaves, but it's actually a gymnosperm. And then you have the ovule, just almost looks like a fruit, but it's not. So here's yew. Again, you might go, is that a fruit? Nope, that's the cone, believe it or not. Yes,
I'm not sure, but they're like green balls and they're like dye your hands. Are those also, you know what I'm talking about by chance?
the junipers probably,
Yeah,
juniper
juniper.
berries. Yeah, we'll get into that next.
Oh, okay.
Yeah. No, that's fine. You're thinking. So the junipers are really interesting because again, if you're just walking by and you look at those, you're like, oh, look at those berries. Can we eat those? It's like, well, you could try, but
I don't,
I would not recommend it.
they are though covered in wild meat. That to me, but
Yeah, but I would, yeah,
don't eat the berries.
no, no, definitely not. They ferment the berries. Do you know what they make out of juniper berries? Gin. Yeah, that's where they get gin from. But their scales, it's a strobilus. The scales are really small and fused, so they look berry-like, but don't eat it. And then the cycads, they have the largest cones. They're huge, 1 meter long, 15 kilograms in weight. So really, when you look at cone tissue, technically it's branches that have been highly modified into the strobili cluster. And the pollen cones are always smaller except for cycads. As far as we know, cycads are the only ones that have their male cones bigger. Otherwise, the female wins. Big eggs, big cones. And when you look at the Pinus genus, they cones get woody, especially the female cones. So there's the juniper berries, but they're not berries. Those are cones, modified compared to what you think about when you think about a pine cone. But that is a strobilus, a cone. That's what the tree can look like. Around here, junipers tend to grow more bush-like. Out west, they tend to be more tree-like. So they can adapt pretty dramatically to their environments. Here's juniper again, and then cypress is another example of really small strobili. And then here's cycads. So on the left is actually the male cone. They're bigger, taller. The females are still impressive though, a little more plump, but in terms of length and weight, the male cone wins on that one. But let's go back to Pinus, that genus. So these are male pollen cones. And all those little dots, those are going to develop into pollen grains. So as you can see, it's a cluster of sporangia. Each one of those, that's sporangia tissue. And inside each of the sporangia, all those little dots, those are what are going to develop into the pollen grains. So you can call it a microstrobulus or a microsporangiate cone, pollen cone, male cone. Okay, but what happens is you'll get those microsporocytes or the microspore mother cells, and they have to go through meiosis. Each one will go through meiosis. Each one produces 4 cells. Each of those survive and develop into the pollen grain. So that's why you see so much pollen in the air during pollination. Okay. So that previous image where each of those microsporocytes have to go through meiosis, and then they're going to all 4 cells stay alive, and each one of those develop into the pollen grain. So when it's shed, we say it's immature. It's an immature microgametophyte. We don't really talk about antheridia in this, really. Plant biologists tend not to say that. We just say it's a microgametophyte. It's so highly modified compared to what we've been thinking about as a microgametophyte. So, eventually we will get a body cell showing up that will divide and produce 2 sperm. They're non-motile, no flagella, no free water is necessary. This is primarily wind mechanism that's going to get pollination to occur.
Gravity. It's like it has to go down the road.
Yeah, I think it's just maybe brownie in motion too. You know, but gravity, I'm sure, plays an impact. Yeah. So these are pollen grains that have been treated with a chemical to get the pollen tube to start to develop. And then they made a microscope slide out of it, just so that they could show the development of the pollen tube. Okay. Yeah, they do, especially this one. Like an elephant. So eventually pollination will be to get to this, right? So these are ovules developing on the female cone. So right here, as you look, each of these are the scales of the pine cone. And then what you have to do is kind of look at the top portion here of each scale. And in this image, we're seeing, one ovule here and here and here. But when you look down into a female pine cone, there's actually 2 ovules per scale. And then 2 seeds will develop per scale. All right. So in this image, of course, you have to imagine there's one right behind this one. Okay. So there's 2 ovules developing on the upper surface of the cone. So each seed scale has 2 ovules, and then it gets fancy. So I'm not going to ask you to know what a subtending bract is or a seed scale complex. Just know that the female cone is a complex tissue. And the one thing I want you to know is that on each scale, there's 2 ovules developing. So springtime, eventually we'll have springtime here. You'll see the scales will be open. Like, okay, pollen, where are you? Okay, they're waiting to be pollinated. So they're waiting for the pollen to come and get delivered. And then you typically will see there'll be a pollination drop at the micropyle region to catch the pollen flying by, and it gets stuck to the pollination droplet. And then what happens is that pollination droplet tends to slowly dehydrate, so to speak, and that causes the droplet to kind of drop in to the inner portion and deliver the pollen in. So it's kind of a neat little system. And then once that happens successfully, then the micropyle will fuse so that the integuments will fuse, so the micropyle goes away. There's no opening anymore. So again, nicely protected because it takes so long for the next stages to play out. Okay, so again, this is all in the PowerPoint slide. You don't have to write it down word for word. I have to move through some of this more quickly. So this is what we're zooming in on. There's one behind it. Here's another ovule. Here's another ovule. Okay. So the pollen got in there, the integument fuses, we have eventually fertilization, and that integument will keep protecting because now that integument becomes the seed coat. So it still has a job to do even after we have successful fertilization and the developing embryo. And then the megagametophyte is the nutrition source of the seed. And once everything's good to go, the cones will separate again. So the cone scales themselves will also fuse during this whole process, not just the integuments on each ovule. You'll tend to get the whole cone fuse. I'll show you pictures. And then again, when the seeds are ready to be released, they'll open up again. And it's usually wind action or animals going after the seeds that will disperse the seeds. They usually have some kind of winged appendage. And then you probably have learned by now in some of them, like BI-112, that some cones need to be going through a fire situation before they'll open up. But that's kind of an exception. But we do have some around here that are like that. We have a lot of jack pine around here. So that's a good example. So here you're looking at blue spruce kind of along the cliff. And if you look up top, that's what they zoomed in on. Over here on the right, you can see all the female cones. So you tend to have female cones, when you look at a sporophyte, a tree, you tend to see the female cones more toward the top of the tree and the male cones more toward the bottom, or sometimes out on the like the tips of the bottom branches. And they have a hypothesis for that. If you've ever been walking in the woods where it's a brisk wind, windy day, you don't really feel an updraft in the woods of wind blowing straight up. If wind was blowing straight up, then the male pollen down here would fertilize its own cones, right? But you don't really feel that. Usually if it's a windy day, it's a side wind. And so the female cones are up top where it's less crowded, and they're waiting for the pollen to come by from another tree. Okay, so they think this design of putting the male cones down, female up, it keeps from self-fertilization. Because in a true forest system, you tend not to get updrafts like that. And so you don't run the risk of self-fertilization. And so they consider that a decent hypothesis for why pine cones, the male and female, are structured on the tree, their locations. So here, there's a couple of ways you could interpret this. Yes. Yeah, you can. I don't know how much has been looked into that. Like, is there a way for the megagametophyte to control which sperm? You know, like, oh, you're one of my pollen. I don't really, and I wouldn't. Yes, and it's usually, if that happens, it's probably a chemical communication or a chemical sensing that's going on, but I don't know how much that's been studied. I know they've looked at it in the angiosperms. So I'm guessing it's the same process that could occur in the gymnosperms too. Well, they still seem to strategize. Yeah.
But like, they don't have it where they start like getting weirdly.
No.
And not like.
No.
being able to function.
No, that's kind of human intervention when we kind of do that. and-
Yeah, that's like human like breeding the plants.
Yeah.
It's like-
So on the left, you see a pine cone that's all fused, right? All the scales are fused. So there's two ways to think about that. Maybe seeds are in there almost ready to go, but they're not quite. And so they're still developing. Or maybe pollination just happened and they just closed up and they're getting ready to do all the rest of it. So there's a couple of ways to think about it. Here, it could be pollination. I'm ready for the pollen, or it could be, I'm releasing my seeds, okay? But this is essentially what you see. And we would have to like look at this one, like it's kind of hanging down right now. If we plucked it off the tree and looked down, we would look for 2 seeds on each scale to know if that's the stage it's in, or is it just waiting for pollination? So we would need to do a little further studying, but I think you get the gist of closed and protected cones versus open and ready for either pollen or release seeds. And then, here with this idea, you need fire to sweep through, and heat is the relevant thing. The heat is needed in order to get scales to open, and that's for seed dispersal usually. Okay. So that's it on that slide set. Let me just introduce the next slide set. Still on gymnosperms. So I won't get too far in it, but let me just introduce a couple of ideas with regard to the needles. Where is it? All right, so if someone brings you a baby sporophyte of a gymnosperm and says, "What is this?" Good luck. Because
It could be anything.
what's weird is that initially all the needles come out in a single, just one needle. They don't bunch. You know, I'm sure you learned if you took BI 112 that there's different bunches of needles, and that's one way to identify, you know, red pine versus white pine and blah, blah, blah. Nope, when they're young, they all look the same in that regard, where they all come out single. And then it takes 1 to 2 years before they finally start to grow in clusters. So then we start using the word fascicle. That's the fancy botany term for clusters of pine needles. And so here you see like there's 2 pine needles, that's red pine. And then here I can't even count how many are in this cluster. Okay, so it takes a couple years before you're gonna see that type of growth pattern. So really young, I can't figure out what's what when they're that young, but eventually you can start counting. So the pine needle, if you take it in cross section, the drawing here is pretty accurate to what you would see. You have sunken or recessed stomata because pine needles last a long time. They go through a full year. They don't drop their needles every year in the fall. At least not all of them. And so they need a little bit of protection from too fast of a transpiration rate. So they kind of tuck their stomata. And that slows transpiration. You see the vascular, it's a pretty narrow, petite leaf, so to speak. So you don't see a whole lot of vascular tissue and internal tissue. Technically, the pine needles aren't true leaves, but I'm okay if we say leaves. I get that, or just say pine needles maybe. So here, what you're seeing on the left is a fascicle that got cut in cross section. Whoops, so you see 3 needles, pine needles in that fascicle, okay? And then they zoomed in on one here on the right, so you can see the tissue, what it really looks like under the microscope, okay? Here's some more pine needles in cross section. You can see the sunken stomata. Well, they're not stomatal crypts, but they are definitely sunken, and that will help slow down transpiration. Okay, so what we're gonna do on Wednesday then is we're going to do a survey of the other 3 phyla. Okay, so the Pinus genus is the only one that the life cycle that we went through, that's the Pinus genus. Okay, that's the only life cycle you need to know. And the phylum Pinophyta definitely are the biggest group. Okay, but we'll go through all of them on Wednesday.